This is part B of this lecture on lipoprotein structure and receptor function. In part A, we covered structure. In part B, we cover receptors. Specifically, we will talk about receptor characteristics and regulation, and then aspects of lipoprotein oxidation and relevance to atherosclerosis. In part A, I introduced the general model of lipoproteins, and here is a picture of a typical LDL particle. There are three important principles. First, cholesterol has two sources. It's both absorbed from the diet and it is made in the liver. The second principle is that wherever LDL goes, so goes cholesterol. Cholesterol and its esters are just a very small part of VLDL and chylomicrons, but a major component of LDL. So LDL is a big target for considering cholesterol metabolism. Finally, the third principle is that elevated levels of serum LDL are very much associated with heart disease and atherosclerosis. The question I'm going to try to focus on and address in this lecture part is how does cholesterol inside of the LDLs know where to go? That is, should it end up in a liver cell, around the serum, or be taken up by peripheral cells, and what happens to it when it gets to one of these sites? The starting point for dietary cholesterol is its uptake from the intestinal lumen, where it is taken up by the enterocyte and by complex mechanisms is packaged with other lipids to make chylomicrons. Specifically, the free cholesterol is acylated by ACAT to make cholesterol esters, and the chylomicrons core contains some cholesterol ester but mostly triglycerides, marked by apoprotein B48 as described in lecture part A. And these lipoprotein particles, or chylomicrons, are transferred to the bloodstream, the lymph system, to be delivered to the liver. As I previously indicated, the bulk of cholesterol metabolism starts with its being packaged in VLDL particles in the liver, and these VLDL particles become LDL particles as they circulate through the capillary endothelial cells and are acted upon by lipoprotein lipase. A very important development in our understanding of cholesterol was the postulate by Michael Brown and Joseph Goldstein of the existence of an LDL receptor on cells. To prove their hypothesis, they looked for patients who might lack such a receptor, and they did so by screening for people with very high serum LDL cholesterol levels. They found a small number of such patients, and they termed them familial hypercholesterolemia patients, noted as FH patients. They then compared the biochemical activities in normals versus such people, and based on the differences, established the notion of an LDL receptor. For this great development, they received the Nobel Prize in Medicine in 1985. The essence of Brown and Goldstein's hypothesis is that many cells contain LDL receptors on their cell surface. These receptors recognize ApoB100, the main apoprotein on LDL surfaces, and these receptors take up the particles by endocytosis, forming endosomes, which fuse with lysosomes, where lysosomal enzymes hydrolyze the proteins to make amino acids and hydrolyze the cholesterol esters to make free cholesterol. That free cholesterol has several resultant functions. First, the free cholesterol down-regulates HMG-CoA reductase, a key enzyme in the synthesis of cholesterol. It upregulates ACAT, which acylates the cholesterol to make cholesterol ester, and it downregulates LDL receptors. We will look at more details of this hypothesis now. In science, it's very difficult to prove unequivocally that a hypothesis is correct. Generally, one carries out experiments that, if they turn out to be consistent with the hypothesis, lend support for it. In a short summary, Brown and Goldstein carried out 6 key experiments, all of which turned out to be consistent with their hypothesis. These 6 experiments are summarized on this slide. In the upper left, in the first panel, they took radiolabeled LDL and looked at whether or not there was surface radioactivity binding to cells as a function of LDL. And in normal patients, shown in circles, there was an appropriate binding function. In FH patients, there was no binding. In experiment B, using the same radiolabeled LDL, they looked at how much was actually taken up into the cells, how much radioactivity. And again, there was a normal binding and uptake for normals and a reduced uptake for FH patients. In experiment C, they looked at the apoprotein that was radiolabeled instead of the LDL lipid, and found that the hydrolysis products of the radiolabeled apoprotein B increased proportionally in normals, but was decreased dramatically in FH patients. In panel D, a similar reaction was looked at for the hydrolysis of radiolabeled cholesterol esters, and again, there was a proportional binding, and then products of cholesterol ester hydrolysis were observed in normals but reduced in patients. In panel E, they homogenized the cells and looked for the HMG-CoA reductase activity in the cells. In this panel, normals in circles decreased dramatically their HMG-CoA reductase activity as the LDL was increased, whereas FH patients showed a high level of HMG-CoA reductase activity, and it did not decrease with increased LDL. Again, consistent with the hypothesis that the LDL particles were not getting to the reductase. And finally, in panel F, the esterification of free cholesterol as one increased the LDL was examined, and that would reflect the ACAT activity, which was increased proportionately in normals but not in patients. Similar LDL receptors exist in the liver and in peripheral cells. Depicted here is the liver and a more detailed picture of the uptake of LDL by LDL receptors that accumulate in coated pits to where there are several such receptors, and several particles can be taken up in what are sometimes termed coated vesicles. You can see the endosome formation, fusion with the lysosomes, and the subsequent effects of the cholesterol that is released, similar to those explained on the previous two slides. As the LDL particles are taken up, the level of LDL in the serum decreases. This slide depicts three effects of increased free cholesterol in the cytosol of cells resulting from the uptake of LDL via the receptor-mediated process. The first effect is the inhibition of HMG-CoA reductase so as to make less cholesterol de novo. The second effect is an increase in ACAT activity to make more cholesterol ester from the free cholesterol. And now I want to add the third effect in more detail, which is the LDL receptor protein translation is inhibited. That is, there are fewer receptors that are made and placed on the surface of the cell. And if you have fewer LDL receptors, then fewer LDL particles are endocytosed. And so the serum LDL and consequently cholesterol levels stays increased. Now we are in a position to look at three different physiological situations. On this slide is shown the normal situation for LDL receptor levels, in which the liver cells are producing VLDL. They are passing through the capillary endothelial cells where lipoprotein lipase liberates free fatty acids from the triglycerides, resulting in intermediate density lipoproteins, IDL, and uptake of those products, often called remnants, by the normal LDL receptors. But some of the intermediate density lipoproteins lose more of their triglyceride to become LDL particles, and they too are taken up by the LDL receptor. The net result is that there is a low level of serum LDL, very little atherosclerosis, and the liver just acts normally, producing a small amount of cholesterol. The second example is the other extreme. What happens in the case of familial hypercholesterolemia? Well, there's no working LDL receptors on the liver. This is a genetic defect. The VLDL is still produced normally, but the resultant LDL is not taken up by the liver and stays in circulation. So one develops a high serum LDL level. It leads to lots of atherosclerosis, and the liver produces its normal amount of cholesterol and is not downregulated. Now let's look at the third case of normal livers, but the individual has a high cholesterol diet, the so-called McDonald's diet, and the cholesterol accumulates in the liver, which does turn down the HMG-CoA synthesis of cholesterol, but it also turns off the synthesis of LDL receptors. So the surface does not contain the LDL receptors. One is making a lot of VLDL, and the LDL accumulates in circulation. So the resultant is an elevated serum LDL and lots of atherosclerosis, even though there's no genetic defect. We just saw that there are two causes of high cholesterol in LDL in circulation. One, high dietary intake, and a second, a genetic lack of the receptors. But there are also many other contributing causes and other genetic components that can lead to higher levels of LDL cholesterol in circulation. What is wrong with high LDL cholesterol? The trouble is that when it is at high levels and sits around, so that serum contains these elevated levels, they're subjected to acetylation and oxidation. And these acetylated and oxidized lipoproteins are taken up by macrophages and other cells by chemotaxis. And these are the cells that end up in the artery in the form of foam cells and fatty streaks, leading to advanced lesions and eventual rupture, thrombosis, and infarction, as introduced at the very beginning of the first part of this lecture A. This is illustrated more explicitly where the LDL classically is taken up by the Brown and Goldstein LDL receptor, and cholesterol synthesis is downregulated. When it has been oxidized, then there are chemotactic receptors, and acetylated LDL is taken up by a so-called scavenger receptor. So there are many kinds of receptors on macrophages, and the oxidized LDL is considered a precursor to the formation of foam cells. I would like to end Part B with the same slide that I started Part A to summarize what I hope you learned about the details that are portrayed in Atherosclerosis Progression, in which macrophages take up LDL by the processes we indicated, especially oxidized and acetylated LDL, which has a propensity to form fatty streaks and foam cells. And with their accumulation, one gets the necrotic core and fibrous cap formation, that eventually leads to rupture, thrombosis, and infarction.